Hello everybody, thank you for joining us in this session today. It's an absolute pleasure to follow Heidi Ream. My name is Nicola Whiffin. I'm a group leader and Sir Henry Dale Fellow at the Wellcome Centre for Human Genetics at the University of Oxford. I'm going to take us into the world of non-coding regions and talk a little bit about how we're beginning to interpret some of the variants in these regions for a role in disease. So as by way of background, the majority of our clinical sequencing currently focuses almost exclusively on regions of the genome that code directly for protein. This strategy makes perfect sense. This is a very small amount of the genome, so around 1.5%, and variants within these regions likely have a very large effect because they directly impact the protein. And also this effect is one crucially that we can predict. We have this beautiful amino acid code that allows us with some accuracy to be able to predict the effect of any single variant on the protein sequence. But through this approach, we currently only find a likely diagnostic variant for around half of all rare disease cases. Now this varies depending on the disease, with it being over 90% in some single gene disorders and much lower, around 20 to 30% for some more heterogeneous diseases. But it's clear that across the board we are missing something here. Outside of these protein coding regions are a wealth of regulatory elements that control the expression of the genes and the resultant proteins. So here I'm showing you a schematic drawn by the wonderful Jamie Ellingford, showing you a few of these elements. So here we have these tall green boxes representing the coding exons that are punctuated by these intronic regions. They are flanked at the 5' and 3' end by these untranslated or UTR regions that form part of the mRNA but not the final protein. We also have a promoter element shown here in blue that drives transcription of the DNA into RNA, and further up and downstream we have these cis regulatory elements, enhancers or repressors, shown in orange. And these regulatory elements sit inside a regulatory system within a topologically associated domain, or a TAD, flanked by these TAD boundaries or insulator elements. There have so far not been very many variants in these non-coding regulatory regions that have been associated with disease. And we can see this by looking at their under ascertainment in the ClinVar database. So here, if I take different regions of the gene and its nearby regulatory region, so the coding sequence in green, intronic sequence in orange, UTRs in pink and purple, and a promoter sequence corresponding to the first two kilobases upstream of the 5' UTR in green, we can see that if we look at the amount of relative genomic footprint of these regions, a large proportion of this corresponds to intronic sequence, with only a small portion being coding sequence. But if we look at the variants within ClinVar, a much larger majority of them, so 75% of them, are derived from coding sequence, with very little being from promoter sequence especially. If we looked at purely the variants that are classified as pathogenic or likely pathogenic, this becomes more stark. So here we see that there are very, very few variants that are not either coding or intronic that are classified as likely pathogenic or pathogenic. So to put some numbers on this, if we go to high confidence pathogenic variants, so ones that have been called as pathogenic by multiple submitters, in ClinVar we see that there are very few in the UTRs and in the promoter, and a higher proportion of these variants are classified as variants of uncertain significance in this data set. So why do we see this under ascertainment? So you could say that variants within non-coding regions are rarely pathogenic. And I would argue that we actually don't know this yet, and it's definitely going to be the case that a smaller proportion of the total number of variants are going to be pathogenic or have large effects. But what is more likely the problem here is that we aren't routinely looking for these variants. We're not routinely sequencing non-coding regions as we're using panel and exome sequencing. And actually, even if we are sequencing, we often are bioinformatically excluding everything but the coding sequence, primarily because we don't know how to interpret the variants that we find within these regions. And this is a self-fulfilling prophecy. So the more that we don't sequence these regions, the fewer variants that we identify, the fewer variants we have to learn from to inform our interpretation, and we repeat this cycle. So most of what we do know from variants in non-coding regions that cause disease come from these single gene disorders. And this is because while we have a large proportion of patients having mutations within a single gene and only a few variants that are missing coding mutations, we can then begin to screen the regulatory regions specifically of that known gene and identify new types of variants that cause disease. However, through some work that we've done recently, we can see that actually this is also the non-coding region variants are also an important cause of more heterogeneous diseases. So, for example, severe developmental disorders. And here I'm just showing you a paper that I'll come back to some of the results from later, where using the Deciphering Developmental Disorders, or DDD study, we showed that for a single gene called MEF2C, nearly a quarter of all variants in that gene that cause disease in this data set are entirely non-coding, so they don't impact the coding sequence at all. But where do we start with trying to interpret these non-coding regions? So the genome is huge, it's 3 billion base pairs, and 98.5% of it is an awful lot of search base. And we know that a large proportion of it has some kind of annotation when we look at things like the ENCODE and the roadmap projects. There's an awful lot of noise, so genome-wide tests for rare variants have incredibly low power, and a large amount of variants within non-coding regions have very little or no effect. And even if they do have an effect, that effect is incredibly hard to define, and it's also very difficult often to define which gene that variant is having an impact on. But how about we start by focusing on regions with very strong effects on genes that we already know cause disease. So going back to our earlier diagram, what I mean is these regions that are either the gene itself or directly adjacent to the gene. So focusing on we can call near-coding regulatory elements. So there's introns, the UTRs, and the promoter sequence, for example. And my favourite of these are UTRs, or untranslated regions. So these are the regions that are directly adjacent to the coding sequence. They form part of the RNA but aren't translated into protein. And they have incredibly important regulatory roles. They regulate the stability of the RNA and where it's trafficked to within the cell, and also the rate at which it's translated into protein. And because of these really important regulatory roles, variants within these regions can potentially have a very large impact. And crucially, in terms of our difficulties with interpreting non-coding region variants, they regulate known genes, they're literally stuck to the sides of them, and they also contain a variety of known functional elements that can help us to begin to predict the effect that the variants are having on gene expression. So for example, here I'm just showing you a figure from a paper that shows the different regulatory elements that RNA biologists and others have found within these untranslated regions at both the 3' and 5' end that regulate these processes. So for example, so here we've got in green the coding sequence, and everything to the left here are elements within the 5' UTR that mainly regulate translation. And to the right of this green box are the elements that exist within the 3' UTR that regulate subcellular localization, RNA stability, and also control translation. So if we disrupt these really important regulatory elements, this can result in an alteration of protein levels, and this can lead to disease. And we recently looked at this question specifically focusing on upstream open reading frames, or UORFs. So these are small elements, as shown by this orange box here, where their start and in-frame stop codons that are encoded actually within the 5' UTR. And these aren't rare elements, they actually occur upstream of around half of all known genes. And a previous study by Kelvo et al. Back in 2009 showed that these individual elements confer a reduction of around 30 to 80%, and this was through testing a handful of examples. So they're common elements, and they confer quite a strong control on a regulation of translation. And they do this because the ribosome, when it binds to the 5' cap of the RNA, it hits the UORF start codon before the coding sequence itself, and that UORF can sequester the ribosomes. It can result in not all of them making it to the coding sequence, and therefore it lowers coding sequence translation. So we recently did some work published in Nature Communications last year, where we looked at what happens if we create one of these UORFs, or create an upstream start codon, or a UAUG, into the 5' UTR. What effect does this have on translation and on in disease? So I've not got time to go through this in detail with you today, but to summarise some of the key findings from this work. So we used the NOMAD database of 15,708 whole genomes. to look at the strength of negative selection acting on classes of upstream AEG creating variants. And this is because variants that are under strong negative selection are not allowed to become common in the general population. So we can see that on average these variants appear rarer. And we found that there is a strong negative selection acting on these upstream AEG creating variants, and this is stronger upstream of known disease genes and haploinsufficient, loss of function intolerant genes, and also that these UAEGs are under stronger negative selection if the resulting ORF would overlap the coding sequence. So instead of having an in-frame stop codon in the 5'UTR, the ORF would actually overlap the coding sequence, and these seem to be more repressive of translation. We also showed that these variants can cause a complete loss of function and lead to severe disease. So just to show you a few examples of that, we looked at variants within ClinVar that are upstream AEG creating variants that explain a large proportion of the known 5'UTR variants in ClinVar. And specifically here I'm showing you, we found multiple variants within NF1 in patients, reported in patients with neurofibromatosis type 1, specifically in a paper by Gareth Evans and team. And in this paper, in a cohort of patients with neurofibromatosis type 1, they identified 4 variants within the 5'UTR. And 2 of these, shown here in red, create new upstream start codons, and 2 of them actually mutate the stop codon of an existing upstream upstream reading frame, and all 4 of these have the same impact, and they lead to a UORF that then overlaps with the coding sequence out of frame and is highly repressive of translation. And given that we know that the effect that these variants have, we might also expect that other mutations within the NF1 5'UTR that have the same impact would also cause disease. And I'm just showing you these here. So there are multiple other possible AEG creating variants across the 5'UTR and multiple different SMVs that would also knock out this stop codon. And these are just the SMVs. There are also multiple different indels that would have similar impacts. So having found that those variants all had the same effect, I actually got in touch with Gareth Evans and said, have you seen any more patients with any of these other different variants that we might predict might be disease causing? And he said that they hadn't, but actually they'd also seen an interesting variant in the 5'UTR of NF2 in a patient with neurofibromatosis type 2. So here I'm showing you another schematic here of the NF2 5'UTR that has this existing UORF shown here in orange. And the variant that we found that Gareth had seen in a patient was a single base insertion in the middle of this UORF up here, this T insertion. And initially this was quite perplexing because this creates an upstream ATG, but this UORF that it creates would be predicted to stop at exactly the same codon as the existing UORF here. But actually what this variant also does is it shifts the frame of this existing UORF sequence, which then bypasses this stop codon, resulting in this UORF overlapping the coding sequence out of frame. And this then becomes a much more repressive and apparently leads to disease in this patient. So because we found that these variants were likely important in disease across different patients, we then created a tool to allow these variants to be detected in different cohorts. So very talented PhD student Xiaolei Zhang created a plugin to the ensemble variant effect predictor that identifies all these different types of mutations that either create or perturb UORFs. So it identifies these UAEG creating variants, variants that disrupt the start codon or stop codon of existing UORFs, variants that shift the frame of existing UORFs, and variants that create early stop codons. And this tool is open source on GitHub. And also we have a publication in Bioinformatics, and this tool in total annotates nearly a third of all likely pathogenic and pathogenic 5'UTR variants in ClinVar. So subsequent to creating this, Caroline Wright from the University of Exeter got in touch and said, oh, should we look for these types of variants in the Deciphering Developmental Disorders or DDD cohort? So here we focused on de novo variants, so high confidence de novo variants identified in a set of just under 10,000 trios in the DDD dataset. We subsetted these variants to those that occurred in 359 genes that had a known haploinsufficient disease role, so heterozygous loss of function mutations. And across these 359 genes, we identified 5 different UAE creating mutations. And all of these are in patients where the phenotype is consistent with haploinsufficiency of that gene. And we were struck by the fact that 4 of these variants actually all occurred in the same gene, and this gene was MEF2C. So this is me coming back to the paper that I discussed earlier. So what are these effects having? So there's actually 2 different effects that these variants are having, and I'm going to briefly explain those now. So in order to look at this, we need to look at the structure of the existing 5' UTR of MEF2C. So this UTR has 2 exons and 2 existing UORFs shown here in green. So 2 of these variants that we found in the DDD cohort create an upstream AEG that creates one of these overlapping out-of-frame UORFs, which I hope I've convinced you by now can be deleterious and cause disease. Another really talented PhD student, Nicola, did some functional validation of these variants by cloning the mutant and wild-type 5' UTR upstream of the luciferase reporter gene, and showed that these patient variants knock down translation of this luciferase gene. The other 2 variants that we found in DDD actually create upstream AGs that are in frame with the coding sequence of MEF2C. So if a translation is dictated from this upstream AEG, it will create an elongated protein extended by 3 and 9 amino acids respectively. And each of these variants we actually observed on 3 separate occasions, all de novo in unrelated patients across the DDD project, a larger data set from GeneDx, and the Genomics England 100,000 Genomes project. So to look into why these variants might be so deleterious, we looked at the structure of the MEF2C protein. And so this is actually the solved crystal structure of the N-terminus of MEF2A, which is identical in sequence to the N-terminus to MEF2C, as it's bound to DNA here in yellow. And you can see from this that the N-terminus of the MEF2 protein, which is a transcription factor, binds directly into the DNA. So you can see here why adding a small amount of sequence to this N-terminal end would completely destabilize this interaction with DNA and disrupt the function of this transcription factor. So Laura Ramos Hernandez out in Madrid did some experimental work for us on these 2 variants, where here we cloned the mutant and wild-type 5' UTRs upstream of the MEF2C gene, and co-transfected this with a luciferase reporter gene driven by expression controlled by MEF2C binding sites. And here we see that compared to transactivation from this assay in wild type, in the presence of these 2 patients' variants, we see much lower transactivation, and showing how these variants disrupt this function of this MEF2C protein. And one of the things that we were struck by when doing this study on the DDD dataset is that we found these 5' UTR variants despite the fact that this data is exome sequencing data. So the 5' UTRs aren't actually included in the capture for this dataset. So just briefly looking at the coverage of this data as we move away from the coding sequence on the x-axis, we can see that there is a reasonable coverage in the regions closest to the coding sequence. And the average 5' UTR is actually less than 200 base pairs long. So for the ones that don't include an intron, as around 2/3 of them, then actually you get quite good coverage of these regions close to the coding sequence. And this has implications for reanalysis of existing exome and panel sequencing data sets, where we might be able to find these UAEG creating variants in different patients for different disease sets. But one of the considerations when we do actually identify these variants is how do we actually interpret them clinically for a role in disease? So our existing guidelines that we use to interpret variants clinically, the variants from the ACMG and AMP, are geared quite heavily towards coding variants. As an example of this, we can look at the PVS1 rule, so the only very strong rule within the guidelines, of which there is also some subsequent guidelines been released by the ClinGen sequence variant interpretation working group. And here we can see the original wording for this criterion, which specifies that null variants are nonsense frameshift canonical splice sites, initiation codon, or single and multi-exon deletions. So as you can tell, these guidelines don't allow for any of these variants being a non-coding a loss of function mutation. However, I hope I've convinced you that some of these 5'UTR variants that we are seeing are very clearly a loss of function mechanism. So to try and tackle the fact that we need some guidance towards interpretation of non-coding variants, we convened an expert panel of this group of wonderful people who have expertise and interest in non-coding variants, with experts in, say, splicing or interpreting enhancer variants, for example. And this was our approach to trying to write a set of guidelines for non-coding region variant interpretation. So we initially drafted some recommendations, going through each rule in detail and deciding how it needed to be adapted to work for non-coding region variants. Then we took this document to this full expert panel, who reviewed it in detail, and we had a series of review calls, and we incorporated their feedback and repeated this process. And that has now been done, and we have a full first draft, which is currently undergoing extensive testing with a group of clinical scientists. So everything I'm going to tell you now is from the provisional set of guidelines, which will be released after we finish this testing process. So first of all, one of the things we wanted to define is actually, when are these variants interpretable in the first place? So we obviously can't go interpreting every single non-coding region variant for every single gene in every single patient. We also can't interpret every single variant within, say, 5 megabases of a gene of interest. So here we kind of very clearly in the guidelines stipulate that we should only clinically be interpreting and reporting variants that fall into regulatory elements that had significant functionally validated links to target genes, and also that those target genes should be clear documented causes of the phenotype of interest. So here we're saying that you should only interpret variants within regulatory regions that are known to be associated with a gene that is on your panel of interest. So this obviously always includes introns and UTRs in a nominal promoter region, but here we're being very careful with wider enhancer regions that they must have a known disease link. What we're not saying is that variants outside of these regions are not interesting. We're just saying that they should be reserved as research variants until some functional data exists to confirm that gene disease link, region gene link. And another thing to note that we discuss in the guidelines is a quick note on terminology. We're very good at referring back to variants as either coding or non-coding, when actually variants that exist in non-coding regions can have very coding effects. So those N-terminal extension variants that I showed you from F2C, for example, are having a very coding impact. It's actually much more useful to define variants based, one, on the specific region in which they occur, so 5'UTR coding exon, 5'UTR intron, for example, and then to refer to the predicted downstream impact. So a variant within the 5'UTR that is predicted to decrease the gene product or to cause a loss of function, as an example. So we then went through every single one of the rules and defined whether they were actually applicable to non-coding region variants or not. So a whole bunch of them are, so I think 13 of the rules are actually applicable to non-coding region variants as they are to any other variant across the genome. So they are completely unchanged in this guidance. For 6 of the rules, they are actually specific rules for types of protein coding variants. So they refer specifically, say, to missense, anonymous, or inframe indels. So these are not actually applicable to non-coding region variants. There was then left a set of 7 rules where we thought that they needed either specific adaption or specific considerations for that rule to be applied when looking at non-coding region variants. Now, I don't have time to go through each of these rules in detail, so I'm just going to specifically pull out one for now. So I'm going to talk to you very quickly about the rule PM1, or mutational hotspot of well-studied functional domain without benign variation. So when we look at, say, either constraint or conservation across regions of non-coding DNA or say, enhancer elements or 5' UTRs, we don't see that conservation or constraint is consistent across the entire region. We see that it is more specific bases that are particularly deleterious when mutated. And because of that, it's not appropriate to say apply PM1 just because we've seen a mutation that is pathogenic in an enhancer before, because another variant in the same enhancer might be having a completely different impact or no impact whatsoever, or a completely opposite impact, either upregulating rather than downregulating gene expression. It is therefore not appropriate to activate PM1 across entire elements. And even when it is appropriate to activate PM1, for example, if it's a very specific transcription factor where multiple pathogenic mutations have a transitive binding site where multiple pathogenic mutations have been seen to hit before, or a specific base that has known pathogenic mutations at it, then we recommend to reduce the strength of this rule to support it, from moderate to supporting, to account for the increased uncertainty in whether this variant is having the same effect. And so that's just an example of how we're trying to refine these guidelines. And we've created this figure, which will look very familiar and at first glance might look like it's the same as the figure from the original ACMG AMP guidelines. But this is the revamped figure that shows only the rules that are applicable to non-coding region variants and has them in modified positions where appropriate, a decrease in strength, and some of them appear twice. So for example, you can see that we don't have a very strong column here because PVS1 is downgraded to a maximum strength of moderate for the non-coding mutations. And as another example, you can see that we have functional data here twice. So we separate functional data for whether it's quantitative in patient-derived tissue and cells, or whether it's non-quantitative and or performed entirely in an artificial system, which has lower evidence towards pathogenicity. And just to clarify here that this is very provisional, because as I said, these variant guidelines are still undergoing testing. But hopefully they will be out over the next month or two when that process has concluded. So just to go on to some concluding thoughts about creating these guidelines and where we are in this space. So we still have an incredibly long way to go. We are only scratching the surface of the different mechanisms by which we know that non-coding mutations can cause disease. And writing guidelines that actually fit all of these different scenarios or predicted scenarios, hypothetical scenarios that we think might exist is actually quite complicated. But I think it is incredibly necessary to try, and we have to start somewhere. And it's only by having these guidelines that we can begin to interpret the variants, to begin to learn more, and to go back and refine these guidance, which I think is going to be a very important step in the years to come. When we have these guidelines fully out after their testing, we really, really value your input into whether they are usable and understandable. And one of the things I'm assuming Heidi has already spoken a lot about data sharing in her talk before mine, but just to say that in this space, data sharing is very important. The only way we're going to learn about these new mechanisms and to be able to help more patients is by sharing the variants that we do find, our predicted impact and our functional data. So please get interpreting and get sharing. So that leaves me just to acknowledge the huge number of people that were involved in different work that I referred to in this talk. So the kind of left three columns here are people that have been involved in the UORF work, throughout all of the UORF work and different papers. And I also hope I've acknowledged people as I've gone through the slides. And on the right here are the people involved in, firstly, writing these non-coding region guidelines. And thank you very much to our testers as well, who are currently reading through these. And obviously to thank the patients that are involved in the GGD study and also Genomics England that we've used throughout this work. And to thank my funders, the Wellcome Trust, Royal Society and the Rose Trees Trust. And thank you everybody else for listening.